Hello friends, subscribe to our channel GDC and press the bell icon for more updates.
Hello Pharma aspirants, welcome to GDC channel. In this video, we will be knowing about the chemical test for glycosides. Glycosides are the organic compounds which on hydrolysis gives one or more sugar moiety along with non-sugar moiety. One is known as glycone and the later one is known as 𝘂𝙬𝘾𝙬. There are a number of glycosides and a number of tests are present to identify each glycoside. First class is anthraquinone glycosides. Anthraquinone glycosides are identified by Bontrager test or modified Bontrager test. In Bontrager test extract is taken and boiled with dilute sulfuric acid. Then this solution is filtered. After filtration, the obtained filtrate is mixed with organic solvent and it is shaken. After separation, upper layer is taken, dilute ammonia is added. This ammonical layer turns into pink or red. Ammonical layer turns into pink or red. This indicates presence of anthraquinone glycosides. In subtype CO glycoside. In Bontrager test extract which is to be identified is mixed with test extract is boiled with dilute sulfuric acid and filtered. The filtrate is added to an organic solvent in a separating funnel. Then it is vigorously shaken. Then the upper layer is removed and mixed with dilute ammonia. This ammonical layer further turns into pink or red color, which indicates the presence of anthraquinone glycosides. Another test is modified Bontrager test. It is similar to Bontrager test but a slight modification is there. In this test, sample which is to be identified is taken and boiled with dilute H2SO4. In additional ferric chloride is added. Then this solution is taken with the presence of chloroform. Then the lower organic layer is separated lower layer, which is organic, is separated and dilute ammonia is added. This ammonical layer turns into pinkish to reddish color, pinkish red in color, indicating presence of anthraquinone glycoside. Indicating presence of anthraquinone glycoside. In subtype C glycoside. In modified Bontrager test, sample is mixed with dilute H2SO4 and ferric chloride. Then the solution is shaken with the presence of chloroform. Then lower layer is separated, which is organic, and then ammonia solution is added, which is diluted. Then the ammonical layer turns into pinkish red color, which indicates the presence of anthraquinone glycoside. Another category of glycoside is cardiac glycoside. Identification test for glycoside which is cardiac is first is 𝙂𝙬𝙎 test. In 𝙂𝙬𝙎 test solution is mixed with solution of sodium picrate. Solution of sodium picrate. In 𝙂𝙬𝙎 test solution is mixed with sodium picrate solution. A yellow or orange color is obtained. Yellow or orange color is obtained, which indicates presence of cardiac glycoside. Indicating presence of cardiac glycoside. This was 𝙂𝙬𝙎 test. Another test is legal test. In legal test, aqueous or alcoholic extract of the drug. Aqueous or alcoholic extract of drug is mixed with pyridine and sodium nitroprusside. Sodium nitroprusside. In observation, red-pink color is formed. Red-pink color is formed, indicating the presence of cardiac glycoside. In legal test, aqueous or alcoholic extract of test solution is mixed with pyridine and sodium nitroprusside, which gives a red-pink color, indicating the presence of cardiac glycoside. Another test for cardiac glycoside is KELLER-KILLANI test. In this test, 1 mg extract is used. In this extract, 10 ml of 70% alcohol. After 3 minutes, extract is taken. This extract is mixed with lead acetate. And in this extract solution, which is mixed with lead acetate, glacial acetic acid and ferric chloride is added, and this is transferred into a tube containing sulfuric acid. Transferred into tube containing sulfuric acid. In Killer-Killani test, 1 mg of extract is mixed with 10 ml of 70% alcohol. Then after 3 minutes, extract is taken out. Then extract is mixed with lead acetate and glacial acetic acid and ferric chloride. This solution is transferred into a test tube containing sulfuric acid. In observation, a reddish brown color appears at the junction of two layers. At the junction of two layers, a reddish brown color appears, in which upper layer shows bluish green color. In Killer-Killani test, a reddish brown color appears at the junction of two layers, in which the upper layer shows bluish green color. This Killer-Killani test is specific for digitoxose sugar and indicates presence of digitoxose sugar. Next test for cardiac glycoside is Raymond test. In Raymond test, glycosidic solution is mixed with 50% of ethanol and 0.1 ml of nitrobenzene. After mixing 0.1 ml of nitrobenzene, 20% of NaOH solution is added, which gives violet color. This Raymond test indicates presence of cardiac glycoside. In Raman test, alkosidic solution is mixed with 50% ethanol and 0.1 ml of nitrobenzene. After this, 20% of NaOH solution is added, which changes the color of solution to violet color, indicating the presence of cardiac glycoside. Another class of glycoside is saponin glycoside. Foam test is performed for saponin glycosides. Saponin glycoside. In foam test, the extract is shaken vigorously with water. Shake the extract vigorously with water. A foam is produced. This foam which is produced indicates the presence of saponin glycoside. Presence of saponin glycoside. Another test is hemolytic test. This test is also for saponin glycoside. Saponin glycoside. In this test, the extract is taken and in the extract, drop of blood is placed on glycoside, placed on a glass slide. Hemolytic zone appears. Hemolytic zone appears, which indicates the presence of saponin glycosides. Presence of saponin glycoside. In hemolytic test, extract is mixed with a drop of blood which is placed on a glass slide. A hemolytic zone appears, which indicates presence of saponin glycoside. Another class of glycoside is triterpenoid saponins. In this test, extract is used which is mixed with acetic anhydride. And this is heated. Lieberman test is a test for triterpenoid saponins in which extract is mixed with acetic anhydride and heated. A pink color appears. Pink color appears, which indicates presence of triterpenoidal saponins. Triterpenoidal saponins. In Lieberman test, extract is taken and mixed with acetic anhydride and heated. Then this heated solution is cooled and concentrated H2SO4 is added, which further gives pink color, indicating the presence of triterpenoid saponin. Another class of glycoside is cyanogenetic glycoside. In this, the first test is test with mercurous nitrate solution. Extract of the solution which have to be identified is taken and 3% aqueous mercurous nitrate solution is added. It gives metallic mercury indicating presence of cyanogenetic glycoside. Cyanogenetic glycoside. Another test is sodium picrate test. In this test, a dried filter paper which was wetted with 10% picric acid and 10% sodium bicarbonate is a filter paper which is already wetted with 10% picric acid and 10% sodium bicarbonate is dried, is placed in the slit of the cork of conical flask containing moist drug powder. A dried filter paper already wet with 10% picric acid and 10% sodium bicarbonate is taken and is placed in the slit of cork of conical flask. This is the conical flask and this is the cork. In the slit with that is gap of this cork, a filter paper which is dried is kept and containing moist drug powder. Flask contains moist drug powder, then filter paper becomes brick or red or maroon color. The filter paper color changes to brick red or maroon color. It indicates presence of cyanogenetic glycoside. Indicate presence of cyanogenetic glycoside. Another type of glycoside is coumarin glycoside. The coumarin glycoside has two tests for its identification. First test is test for Sulleria. In this test, hydroalcoholic solution of Sullerin is mixed with propylene glycol and acetic acid. When hydroalcoholic solution of Sullerin is mixed with propylene glycol or acetic acid, a blue or green fluorescence is seen. Blue or green fluorescence is seen. It indicates presence of coumarin glycoside. Presence of coumarin glycoside and presence of Sulleria. Another test for coumarin glycoside is test for Sulleria. In this test, the alcoholic solution of Sulleria is mixed with sodium hydroxide solution. A yellow color fluorescence is seen. Yellow fluorescence is seen, which indicates the presence of coumarin glycosides. Presence of coumarin glycoside, which is Sulleria. So there was two tests for coumarin glycoside. Test for Sulleria, in which hydroalcoholic solution of Sullerin and propylene glycol mixed with acetic acid gives blue or green fluorescence indicating presence of Sulleria, and in another test, alcoholic solution of Sulleria mixed with NaOH gives yellow fluorescence indicating presence of coumarin glycoside. That's all for the chemical test for glycosides. Thank you for watching this video.